Okay, so I'm going to talk about these VUs, right, that are causing all those problems. And do I have a pointer? Okay, so most but not all genetic diseases caused by missense mutation that causes proteins to misfold, which leads to their degradation in cells, right? Not all mutations, right, but most. For example, Hopwood's lab, the guy that did all the work on LSDs over the many years, 25 years ago, he showed that almost in all LSD patients, if you just look at the abundance of the lysosomal enzyme, it's usually gone just with an immunoassay. So it's mostly degraded, not always. So we think activity is a little better, but not much. Most of the time when you have a genetic disease, the protein is essentially gone because it's misfolded, not always. So it's well known if we sequence genes that are relevant to genetic disease, we encounter variants of unknown significance, the so-called VUs. And when these VUs are rare, we cannot know a priori if they drive a disease. So whole genome newborn sequencing pilot programs announce that VUs will be ignored. I have a lot. Okay, so the newborn screening pilot programs that'll be doing newborn sequencing first here announced that they will ignore VUs, right? They sort of have to. And this will lead to a large number of false negatives. And this is a serious problem, but you could argue it's better to screen for a lot of diseases and catch some than it is to not screen for these diseases and catch none, okay? But, false negatives, we should try to reduce those to zero. For example, for MLD, we put all of the VUs in 1 million humans that we found in the databases into human cells, and 20% of them have no activity. So 20% of the VUs will be MLD drivers, and this will lead to some false negatives. This is published. Okay. So as we move to incorporate first-year whole genome sequencing for newborn screening, it becomes important to biochemically evaluate VUs with new high-throughput technology. VU annotation is also important to support second-tier gene sequencing that is often done after a first-year biochemical test. So we heard a lot today about VUs and inconclusive genotypes, right? And following patients with 2 to 10 nanomolar cytosine that are at risk for late-onset disease. Just consider Krabbe disease as an example. We do first-tier GALC enzyme in blood spots. Then we do second-tier measurement of cytosine. When GALC is low and cytosine is elevated, but not to the extent prognostic for early disease, we go to third-tier genotyping and often encounter VUs. We try to use genotype for prognosis to plan patient follow-up and treatment scheduling, but sometimes we cannot because of VUs, right? To establish, the established way to biochemically annotate VUs is to study them one by one by making the variant in the gene, putting it into human cells, and measuring the function of the protein. Today, I want to talk to you about a new technology that my lab developed that does this in a novel, massively parallel fashion. We call this voodoo because we're going to be doing lots of VUs, okay? We thought about calling it Voodoo Child if you're a Jimi Hendrix fan when it's applied to newborns, but anyways. Okay, so it's kind of like Survivor Island, or maybe like the White Lotus. So passengers board a ship and we record it to a manifest. The passengers do their thing on the island for several days, and then they go back on the ship and we take roll call, and some of them are missing. Okay, so what we're going to do is we're going to put lots of VUs into cells, let them do their thing, and then figure out who's missing because they're degraded and misfolded. That's the idea. So let's talk about missense variant libraries. There's sort of two kinds. One is we can take, we have about 2.5 million human genomes sequenced now, publicly available. I mentioned that earlier to Joanne today. So we can take all the rare VUs in this 2.5 million people and put them into our library, okay? And anything not in that library is going to be an exceptionally rare VU, we think. Assuming that the libraries cover all ethnic backgrounds, which more and more they do. So we'll get most of them. The other thing I'll talk about is a saturation library where we put all possible missense variants into a protein. All possible means each amino acid changed to 19 different other options, okay? One per protein. So every possible missense mutation astronomically possible on the universe will be studied. There's nothing more to study. So the way we do this is, I talked about this a little bit last year. I'm going to show you some initial results and to tell you that we're going to do this for Krabbe disease by the end of the year. So we have a transfer plasmid. We have a variant library, which I told you, saturation library, or the 2.5 million library. And then we put a small DNA in there called a barcode, which is 4 billion 16 nucleotide sequences. So any given variant will have a unique barcode because the number of barcodes is huge compared to the number of variants. So no two variants will have the same barcode. It's that simple. Okay. The barcode, it tells you what the variant is. Okay. We do something called bottlenecking. Just try to get your head around this. It's pretty cool. We don't want millions of barcodes per variant. We want about 50. So what we do is we take the plasmids, we put them into bacteria. We take an aliquot of the solution, put it on an agar plate, and count the number of bacterial colonies. Each bacteria is one plasmid. Okay, so let's say we get 50 colonies, and that's 0.01% of our mixture. So we have 50 times 100. We have this many plasmids. So we want the number of plasmids divided by the number of variants to be about 50. And then statistically, we'll have about 50 barcodes per variant. Try to think about that. It's not that complicated. Okay. But each barcode, you tell me the barcode, I tell you the variant, but we don't need thousands of barcodes for each variant. 50 is good enough. We do this by bottlenecking. Okay, so now here comes the magic. We put this into human 293 cells, the transfer plasmid. I'm going to give you two more elements now. It has a puromycin. This allows the cells to grow in the presence of the antibiotic puromycin. And then it has this BXB1 bacterial integrase site. It's like an HIV thing, but bacteria. In the chromosome of these cells, in one spot on one chromosome, not on the sister, is a BXB acceptor site. The BXB integrase, an enzyme that's encoded in these cells, will insert the entire transfer plasmid into the chromosome at a single site called the landing pad. And once it goes in, it's irreversible. Another one cannot go in. So whichever variant goes into that cell only expresses that variant and none other. Every cell expresses only one variant, guaranteed. Furthermore, the promoter in orange that drives the expression of the variant gene is in the landing pad. So the variant gene only turns into protein once it's integrated into the chromosome and put next to its promoter. Okay, so every cell expresses one variant protein, guaranteed. So there's three versions of Voodoo. Voodoo enzyme is being applied to GALC for Krabbe disease. That's what we're starting to do that. We'll have the results by the end of the year. So we put 2,000 GALC VUs into 293 cells at one variant per cell. These are all the VUs that we can find in 2.5 million humans. Okay, so anything else beyond this, there might be some. They'll be exceptionally rare. So what we do is we use a technique called PacBio sequencing. It's an amazing technique that gives you long DNA sequencing reads. You can sequence a single molecule. We sequence 3 million of these plasmids. So now we have the phone book. It tells us the variant and which barcodes link to it into the same plasmid. Okay, because we sequence it in one go, 3 million times. So give me the barcode, I give you the variant. It's that simple. We have the record. So we know the manifest going into Survivor Island. Okay. We take one tube of cells, and we transfect with this mixture of plasmids, okay, with thousands of variants. And we then sort the cells using a fax machine We put one cell in every well. We have 2,000 variants, so we make 10,000 wells to ensure that every variant is in a well. And we let them grow up, so every well comes from a single cell. And we use a high throughput mass spec assay of GALC to measure the enzyme activity in these 10,000 cells, variants, colonies. And we focus on all the low activity variants, let's say those that are less than 10%, okay? Because if you have more than 10% GALC, we think you could not possibly contribute to even late onset Krabbe disease. We then simply read all their barcodes and we know all the variants in the 2000 that cause less than 10% GALC activity. We can now study these cells individually. For example, we can study, is the protein secreted? Lysosomal proteins are go to lysosomes. Sometimes they miss target, they get secreted. So we can tell not only is the protein misfolded or degraded, but also if it's mistrafficked and does not go to the lysosome. We can do a number of biochemical tests to really study in more detail each of these variants that reduce the activity. And I'll be able to give you a list of all the VUs that reduce the activity to the point that you need to worry about. Okay. Now let me talk about green voodoo, which we're using for MLD. Now we move to saturation library. All possible missense variants in the universe for this protein are included in the library. And the protein is produced from the landing pad it's fused to a green fluorescent protein. It's green, right? You know, the Nobel Prize for the jellyfish protein that allows you to look at proteins inside cells because they're fluorescently green. If the protein is degraded, it loses its green fluorescence. So here on the left, on the right, you have a red fluorescent protein called M-cherry that's expressed everywhere in the cell. You can see that. In the left, you have the green ARSA enzyme that goes to lysosomes. You can see it goes into these punctate domains, which are lysosomes when we track it with a co-stain with a lysosomal marker. Okay, so this we use for proteins that go to lysosomes, okay? We take these cells and we put them in a fax machine, one by one, boom, boom, boom. And we measure the green-red ratio. It's very simple. If the green-red ratio is low, it means the green did not survive and the protein misfolded. We grab all the cells that have low green-red ratio and we focus on those. And we read their barcodes and we identify all the mutants that cause misfolding and degradation of the green protein. It's that simple. Let me show you that this works. In green, you have the wild type. This is the on the x-axis, you have the green-red ratio, okay? You can see the green peak has higher ratio than the gray and the pink peak. And on the y-axis, you have the number of cells read by the fax machine. So the green is the wild type protein, and the gray and the pink are two mutations that we previously knew misfold the protein and cause its degradation. And you can see that they peak at much lower green-red ratio. There's a little bit of overlap between the green tail and the pink-gray. It doesn't matter because we're counting barcodes. Doesn't matter. We read all this. Remember, every protein has 50 barcodes. So by looking at the green-red ratio and the number of barcodes in each bin, we can tell exactly the extent of protein folding, exactly, quantitatively. And let me end with full voodoo. This we use for proteins that go to the cytosol. We have to use green voodoo for proteins that target to organelles or secreted proteins, but full-on voodoo works for any protein in the cytosol. Here we fuse the variant library to something called MCP. What is MCP? This technique is really cool. We have the protein variant. On the C-terminus is a little peptide tag that we can pull down this protein with an antibody that binds selectively to that peptide. On the left side of the protein, on the N-terminus, we have MCP, which is a bacterial protein that binds a double-stranded RNA hairpin. The barcode in this case is made as a mini RNA. We circularize it inside cells so that it's not degraded. You know, single-stranded RNA is degraded in cells, but we circularize it using a ligase inside these cells that makes a circular RNA that's not degraded. It has the barcode in it, which is in pink, and it has the RNA hairpin that binds to MCP. So if this protein folds and is not degraded, it will capture its own barcode, even though it's not attached to a covalent. And if it doesn't fold, it will under capture its barcode. So we lyse the cells, we bring down all the proteins with the little yellow tag, and we sequence all the barcodes, using Illumina sequencing. And that's the patient, that's the manifest of patients that got on the ship at the end of Survivor Island. We compare that to the manifest that got on the island, and we know who got killed, right? Who's missing. All right. I think I just said all this. Just finished. So what are we applying voodoo to? Aldolase B, hereditary fructose intolerance, very common disease, super easily treatable by lowering fructose. Nobody has a good screen for it. We might be doing it by DNA. We want to map out all the VUs in aldolase B. For the green voodoo, we're doing ARSA for MLD, and for Krabbe disease, we're doing voodoo enzyme. This just shows you that voodoo works. We did this on a library of 12,000 mutants of a protein called BRAS for a cancer study. And when we measure the voodoo score versus the Western blot score of protein abundance, they agree perfectly, showing that the method works. This is published, by the way. And this is the voodoo team. So I'll just leave you with that. Thank you.